Hi, my name is Kate Levinson, and today I'm going to be discussing the genetic code and the experiments conducted to conform the genetic code. The genetic code is a table that displays the relationship that mRNA, in the form of codons, has with amino acids. A codon is a set of 3 nucleotides of mRNA that form a unit in the genetic code. Nucleotides in the genetic code are adenine, guanine, cytosine, and uracil. So the first experiment we're going to talk about today is Nirenberg and Matthaei. These two scientists were the first to determine that a specific sequence of RNA will code for a specific amino acid in 1961. The two scientists added a homopolymer, which in this case was poly-U, into 20 test tubes containing a cell-free translation system and 20 amino acids. In each of the tubes, 19 of the amino acids were unlabeled, while one was radioactively labeled. The experiment was repeated 20 times, each time with a different amino acid radioactively labeled. Once the homopolymer was in each of the tubes, the test tube was incubated and translation occurred. They then checked for radioactivity. If the labeled amino acid produced a radioactive protein, a protein chain was forced of one amino acid. For them, the breakthrough came when poly-U was discovered to encode for the amino acid phenylalanine. They also discovered that AAA encoded for lysine, CCC encoded for proline, and GGG did not encode for an amino acid. The second experiment is Nirenberg and later. They wanted to determine other combinations of codons that encode for different amino acids. This experiment began in 1964. The experiment began with short sequences of mRNA, which is also known as messenger RNA, and they were synthesized and combined with a mixture of ribosomes and amino acids that had tRNAs attached. The ribosome connected the mRNA to the specific tRNA that matched the codon. The solution was filtered, and the bounded tRNA and mRNA was caught in the filter, while the unbounded tRNA fell through. The scientists assayed the filter and were able to determine which amino acid was bound. When the mRNA GUU was used, the tRNA, which was bound to valine and read CAA, were found on the filter. The result of this was that the codon GUU encodes for the amino acid valine. The pair was able to identify 61 other codon and amino acid relationships. Overall, these two experiments helped progress the understanding of genetics as well as set a basis for the production of the genetic coding dictionary. The coding dictionary can be seen to the right.